At the end of the last lecture on energy metabolism, we talked about that ATP is the thing that does cellular work. So what I did is I decided to make a separate lecture on ATP so that one didn't get too long. So here we go. ATP. It is adenosine triphosphate. So like the name might give you a hint about, there are 3 phosphate groups connected to an adenine. And there's a ribose sugar in the middle. So here's the triphosphate, here's the sugar, and then here's an adenine, okay, which is a nucleic acid, right? One of our nucleotides, okay? And so that's the structure as is when it's ATP, okay? So here's what happens. When you break down ATP, it gets broken down by hydrolysis. We learned about that in Unit 1. Remember water coming in to bust a thing apart? So basically what water does is water comes in right here, and it's going to break off this last end phosphate group, which is going to leave our adenine, our ribose, and 2 phosphates, which is called ADP for adenosine diphosphate, di meaning 2, okay? So when water comes in, it breaks off this one, and now this one is over here. When it's free floating, it ends up usually being referred to as PI, inorganic phosphate, because now it's not attached to one of our molecules of life, okay? And here's the ADP. As we know from hydrolysis, this releases energy. It's exergonic, and so here's the energy over here, okay? So when you break down ATP, it ends up being that the delta G is negative 7.3. Don't worry about these units, okay? So this is spontaneous, it's catabolic, it's exergonic. So this is energetically favorable for ATP to break down, which is great because your cells need to break it down a lot, okay? So remember that there are 3 different kinds of work that your cells can do: chemical work, transport work, and mechanical work. So we're going to now take a closer look at how ATP can help your cells do those different things. Okay, let's start with chemical work, which is maybe kind of the most complicated one. The chemical work that your cells need to do is, remember back how some reactions are energetically favorable and spontaneous and some reactions aren't? Like those anabolic reactions are not favorable. That's the work that your cell has to do. Okay, so anything that's not going to happen on its own, anything that requires an energy input, ATP is going to be used. Okay, and so what happens is we're going to break down ATP and we're going to release some energy, and that energy that's been released is going to be used to do that other energetically non-favorable reaction. Okay, this is called energy coupling. Use the energy released from breaking down ATP to do a reaction that wouldn't normally happen. Okay, so you like pair them up together. So I think this is best seen by an example. All right, so here is an example. This is an anabolic reaction to make glutamine, which is an important amino acid that we need. Okay, and so in order to make glutamine, you've got to take this stuff called glutamic acid and you have to add ammonia to it. Okay, but this reaction, look at this positive delta G, which means it needs an energy input, so it's not going to happen on its own. Okay, so here's where ATP is going to come in. So what happens is ATP gets hydrolyzed. Okay, it's going to break apart. And one of the things that's going to happen is, notice now we've got that ADP and that free-floating phosphate. It's going to attach itself to this glutamic acid. And that might not seem like that big of a deal, but when it does that, that makes this guy kind of like unstable. And that instability is going to allow ammonia to come in and attach to it, whereas ammonia couldn't do it when it was in this form. This is too stable, but now just adding this phosphate group makes it unstable and makes ammonia able to come in and bond with it. Okay, so basically the ATP breaks apart, releases energy. Okay, it tacks a little phosphate on there, which makes this unstable, and now this ammonia can come in and bond, and here's our product, and now we've got that free-floating inorganic phosphate group again. Okay, so remember that this reaction, the general reaction, is overall positive, but notice that when we split ATP, it releases 7.3. This requires 3.4. So as you can see, if you kind of take these and couple them together, you end up having a thing with a negative delta G, which means it's spontaneous. So this is spontaneous as long as ATP is present. Boom, the energy release from the ATP is going to help this other reaction actually happen. So it happens because of the energy release and because of this phosphorylation makes this thing unstable, okay? So your cells do this all the time. This is the only way that these unfavorable non-spontaneous reactions can happen is through the breaking down of ATP. One thing you should know though, like we talked about metabolic pathways, sometimes what you make is the beginning to something else. Okay, and it kind of keeps going in a chain. And this ends up being a much more efficient way for your body to process things and transfer energy. So that was chemical work. So your cells use ATP and energy coupling to do chemical work. What about transport work? When I'm talking about transport, we're talking about transport across the cell membrane. Okay, so notice here we've got a transport protein. And when ATP is hydrolyzed, it's, split off with water. There's going to be some energy released. And look at now what's here. Oh, it's a phosphate group. And this phosphate group, when it attaches to here, is going to cause this protein to change its shape. And so now that solute that was, let me see, inside of the cell now moves bloop outside of the cell. So basically this phosphate group is what opens and closes this like protein door. Okay. So what kind of transport would use energy and like a protein to do that? Active transport. That's right. So that's how your cells use ATP to do transport work. Now mechanical work. This is when things move. And so here's an example. There are some kind of tracks in your cytoskeleton, like inside the cytoplasm, that things can zip around in, kind of like little roads in the cytoplasm. Okay, and basically if you want to move a vesicle along the roads in your cell, you're going to use ATP. And so again, this ATP, when it's split apart, is going to attach a phosphate here, and that energy and that phosphate attach is going to make the vesicle go along this little track. Okay, so it causes movement here. So I don't want you to get super bogged down in the details of this, but I want you to understand that when ATP is split apart, it releases energy and it phosphorylates something. That means it adds a phosphate group to something. And when you add a phosphate group to something, that's going to have that thing do something different. Okay, it's going to allow it to do some kind of work it couldn't do before. So as we know, we can take ATP, we can add a water molecule and break it apart into ADP and then that free floating P. But then how do we make ATP? Well, basically that free floating P and that ADP get joined back together. This is going to require an energy input from some other reaction that releases energy. We're going to take that energy and use it to connect the ADP and the P back together. And so now we'll end up with ATP again, and it'll get it broken down, and then we'll take some energy input and we'll make it again and break it down and make it again and break it down and make it again. Okay, so this happens crazy fast. Okay, like this graphic over here says, a muscle cell has about 10 million molecules of ATP, and it can recycle them all in 1 second. That's how much it uses, but that's how fast it can regenerate too. If we weren't able to regenerate our ATP, we would have to basically eat our body's weight of ATP every day. That's how much ATP your cells go through in a day. Okay, so this is constantly recycling, constantly taking energy from some exergonic reaction, using it to rebuild ATP. This process is called cellular respiration. We'll talk about that later. And then we can break it down to do other things over and over and over and over and over again. Here are the main highlights of this lecture, and here's where we are in the book, chapter 6.3.